So we have been fortunate to have sponsorship for this symposium. And the sponsorship came from the Agron Institute and the Alfred P. Sloan Foundation. I wrote them a couple of grants. They gave us money. It allows us to do this event, which is wonderful. And I want to make a statement here from Paula at the Alfred P. Sloan Foundation. She says, Norm, I'm so sorry I cannot be here to celebrate all your remarkable achievements. Thank you for your leadership, vision, and guidance. You helped create Sloan's Microbiology of the Built Environment program, and your work on showerheads, subways, drinking water systems, and more has been critical to the success of the program. We truly appreciate all you've done, and we hope that your retirement will include a lifetime to occasionally help the Sloan Foundation. We are immensely grateful. And then Joan Kabori at the Agron Institute emailed me last night, and she said the webcasting was great. She wants us all to enjoy this event, and she thinks it's an amazing time to celebrate some remarkable achievements. So those are what our sponsors say, and that's what our sponsors think, which is great. And I think we'll get going for today.
I'll just note in passing, the Sloan Foundation support has just been really wonderful. All of them, really strong support. And in part, the reason we were able to get money for this is because of you, John. You're heavily involved in agro over the years. So, yeah. So thank you, John.
You guys want to go? Yeah. Zach. So today, the way we're going to do this is that each speaker is going to have one of the School of Mines students introduced. And this will be Zach Jones.
Hi everyone, thank you for joining us this morning. My name is Zach Jones. I'm a PhD student here at the Colorado School of Mines, and I work under Josh Sharp. We work in the same lab as John, so it's a microbiome lab. My research focuses on studying wetlands, and I look at the wetlands and look at the microbiome and trying to see how it relates back to nutrient cycling and pollutant removal. Today I have the pleasure to introduce Dr. Daniel Frank, who is a professor at the University of Colorado Denver and Schultz Medical Campus. Dr. Frank received his BS in biochemistry and biology from the University of Illinois, and a PhD in biochemistry and biophysics from the University of California, San Francisco. While working under Dr. Pace, he studied RNAsP and began research on the human microbiome. Dr. Frank is the creator of Explore-seq software used for phylogenetic analysis of sequence data. His research now focuses on ultra-independent techniques to study how human hosts and their associated communities interact to contribute to mother-child HIV transmission, Staph aureus infection, and inflammatory bowel disease. Please join me in welcoming Dr. Frank.
Thank you for inviting me. It's an honor to be here. So I'm going to kind of continue Norm's theme of talking a little bit about the history of how we got here, got to the point where my lab is. I started out, did my PhD in Chris Guthrie's lab many years ago to focus on understanding the RNA, small RNA proteins that were involved in pre-messenger RNA splicing. There was a lot of in vitro splicing work, some structure function studies of small nuclear RNAs, looking at the spliceosome using molecular genetics, very complex dynamic systems. And so it was time to choose a postdoc. I'd already seen Norm at various of the Cold Spring Harbor meetings and interacted with him a bit. And I thought that would be a great lab. I was very interested in going from taking maybe a little more reductionist approach to studying RNA enzymology. And so as Norm mentioned yesterday, I came in the lab as an RNA jock working on RNAsP, doing in vitro selections, a little bit of eukaryotic RNAsP. I get tickled by this poster, this picture, because if I saw any of my students now working without their lab coats, their coffee mug over here, I'd probably fire them. But it was a fun time, but I quickly realized, I'm sure Norm realized before I did, that I'm not a chemist. I'm not going to be the guy that speaks Russian and crystallized proteins and RNAs and such. And so at that time, Norm said, Dan, come to the dark side, study this microbial diversity stuff. And I dabbled a little bit of that as an undergrad, so I said, yeah, that sounds interesting, let's do that. So that's how I got into, many years ago, working on microbial diversity. Now, the rest of the day...
Can I interject?
Yes.
Dan, by the way, spent time with Carl Woes at the University of Illinois, so it's a very special connection.
Thanks. And so the rest of the day, you'll hear about all these marvelous places people are going to go to sample, collect specimens, or at least go on boat rides and such. I still wonder why Phil's peeing into this hot spring. The environment I got to study is this: the small and the large intestine of humans. And so when John talks about people passing through the lab, I have the honor of studying what's being passing through these complex environments. And you can imagine that there's all these little interesting nooks and crannies. There's going to be some interesting microbiology going there too. And so that really, along with other folks, we began studying human microbial ecology. The kind of questions we're still interested in, still developing, are similar to what all of you who are out there Yellowstone get to study. We're interested in relationships. So what do the microbes do for us? We're colonized by about 100 trillion microbes at any time, from birth until death. What are we doing for them? Organizational. What kind of microorganisms are typically present on us? Is there a core microbiome that we're shared by everyone, or is it just kind of random based on who we're interacting with, what we ate, things like that? Got a lot of different niches in our body, so it's the spatial distribution. And then dynamics. So how do these communities develop? We're born relatively sterile under normal conditions and rapidly develop a complex microbiome. Is that driven again by environment or other factors? And we're studying that in the context of things like maternal programming and what C-sections do. How does the host and the environment impact the microbiome? So are you getting influence from your parents, from your kids, or from your siblings, or what? And especially coming at a medical school, we're really interested in how disturbances in these communities really, whether they contribute and how they contribute to disease. And so the way I think about this is, as a host, we're kind of surrounded by this cloud of microbes, and all of our interactions with the environment are basically refracted through that. And so how this cloud forms and how it leads us through life is of interest to me. And the rest of the field, look at human microbiome. And so, like other environments, these are very complex. Fecal contents have up to 10 to the 11th cells per gram of material, thousands of species, tens of thousands of species if we look across the human race, a lot of symbiotic and syntrophic relationships, and that really makes it difficult to do co-postulus type of studies where we isolate one organism, study what it does, and then figure out its role in disease. And unfortunately, through the work of Carl and Norm, which we heard about last night, we now have incisive molecular techniques and a framework where we can now pull out sequences from an environment, tie them into this tree, and infer what kinds of organisms are present in a particular environment at a particular time. Now, a couple of the very first seminal studies came out of Dave Relman's lab and Stan Falco's, where they were applying these techniques to a couple idiopathic diseases where there was good suspicion that there was a microorganism present, a pathogen present in a tissue that just could not be cultured. And so back in the early 90s, they published a couple of seminal papers looking at bacillary angiomatosis and Whipple's disease, where they could see the bugs and then apply 16S techniques and treat the organisms. And Tom Schmidt was involved in both of these, so I think that from ground floors there were various PACE Lab alums at least involved in driving the expansion of studies of the human microbiome.
And I make just a little and this is Relman has been following Relman's lab regarding this stuff. And at one point in the game, I got a call from Relman, and he said, we got these ribosomal RNA sequences. what do we do now? And at that time, Tom was just getting ready to go off and start his own life. And I said, talk to this guy.
And I tell that story, doesn't that say so much about Norm? How many of us today would have said, screw it, I'm going to publish this, I'm going to, I'll be on those papers, a couple New England papers, Seminole papers, where he said, here's a junior colleague. We've all been in that position where we're kind of, Norm was putting us on his shoulders, definitely, without taking this proper credit. So I think at this point, there's this movement thinking.
By the way, when this came up, the first thing that occurred to me was Norm Pace should have been an author on that. I'm disappointed that it didn't work out that way. Because I did that work because you had the facts, and Gary Olson was there and had it set up and run it, so you really should have been
So, at this time, I think we saw this kind of, well, there are all these going to be interesting organisms, pathogens out there. We can discover new classes of pathogens, new domains of pathogens, and I think that hasn't really turned out as much as something that I think is just as interesting, which is rather than discovering new pathogens, what we're finding is many diseases are instead caused by imbalances in the microbiome. The term for this is dysbiosis. So it's the community as pathogen, as Norm termed it, that's dysfunctional rather than a particular organism. So it's the loss or the dysfunction of the bugs that are driving disease. And this has been reported in multiple diseases. We'll talk about inflammatory bowel disease, obesity, C. diff is a real classic case of that. A lot of chronic diseases. And it's always important, have been associated with dysbiosis, but it's always important to think about causal relationships. So if you're treating someone with inflammatory bowel disease with drugs, steroids for many years, it's maybe not surprising that their microbiome is screwed up. It really doesn't lead to disease. So I think that's a challenge in the field, is getting those causal relationships down. But so I was drawn into this in about 1999, sick of RNA-HP, and Norm at that time was brought into a NIAID symposium trying to look at what the causative agents are of inflammatory bowel disease, specifically Crohn's disease. And there's basically two forms: Crohn's, which affects the entire bowel, mainly the ileum; ulcerative colitis affects the colon, and both have really complex etiologies. So there have been probably now about 70 susceptibility loci identified. Mouse models indicate that the microbiome does exacerbate colitis, so germ-free mice don't develop the symptoms of colitis as connotation. We were brought into it because at the time there was a hypothesis that was kind of a religious battle, whether Mycobacterium avium paratuberculosis might be involved in Crohn's. I won't go into the details of that, but that became worrisome because this bug is found, is shed from dairy cattle, and it's not killed by traditional pasteurization. So it's in our milk supply. A lot of cattle have a disease called Young's disease, which are just pumping out tons of this mycobacteria. It's in our milk supply. It's causing Crohn's disease. People were really worried. And so I think Norm was brought into the project to try to understand what could we do to apply our techniques to test this hypothesis. And at the time, 1999, the Enteric Pathogens Research Unit, which was funded by NIH, set up a study where we were collecting full resected tissues from people undergoing surgery for either Crohn's, ulcerative colitis, or other non-IBD diseases. Three of the chunks of each tissue were going to the University of Central Florida or University of Maryland, Michelle Trux's lab, and these were pros at growing mycobacteria. Another chunk was Ed Bodeker, who was an E. coli expert, because there were also hypotheses that E. coli was involved in disease pathogenesis. And just, I guess, because there's a fourth chunk left over, they decided to send that to the University of Colorado, where we could do, mainly we were interested in interested in us doing qPCR for MAP, and then some of the fish studies. And that also gave us a chance to begin applying 16S technologies to these samples. And so we then started this project, which multiple years, where we collected about 190 surgical specimens from Mount Sinai, from a variety of patients. At the time, we had samples from the colon, small intestine, a few cecum, a few lymph nodes. And there's what we tried to do from the Crohn's patients too is pull out tissues that were both from grossly pathological sections and normal sections of the bowel where they're hooking up the bowel after they've resected it. At the time, high throughput was cloning and sequencing, and so we did a broad range bacterial RNA clone libraries using 804, 805 reverse, get about half the gene out. This was difficult because we're looking at resected tissue, so there's a huge amount of human DNA compared to the amount of bacterial DNA. And so the full-length sequences just weren't, PCR just wasn't efficient. We screened a whopping 96 clones per sample, which was huge at the time, and generated a data set of 15,000 Sanger sequences, which was probably one of the larger data sets at that time, certainly the number of patients. And what we found is, what I'm showing here then is just the relative abundances of some of the primary phyla and some of the subphyla of firmicutes we see. And we did notice, first of all, there were differences between the colon and small intestine as we suspected, and we did see significant differences, but minor, between the occurrence of, say, lactospiraceae and either Crohn's disease or non-inflammatory bowel disease in both the small intestine and the colon. And some of these have been, this kind of corroborates some of the smaller studies that had already been published, showing that there were differences in Crohn's. However, I think we were kind of once we started doing kind of ordination techniques as a principal coordinates plot, we were struck by the fact that maybe Crohn's disease isn't a monolithic kind of entity, so we were assuming in these analyses that all Crohn's disease will have a similar microbiome, and it may or may not be different from the normals. And what all the various exploratory techniques all pointed to the fact that there was a subset of the Crohn's and ulcerative colitis patients that seemed to have a dysbiotic phenotype, whereas a large proportion in all the normals and then about half of the Crohn's and ulcerative colitis actually had what we consider a normal microbiome. And if you look then at kind of the stratigraphy of these particular phyla throughout the length of the bowel, you can see that there were profound, in these patients out here, subjects out here, profound disturbances in the microbiome. So up in the ileum, there's just a flourishing of proteobacteria at the expense of the bugs we typically want to be there, such as the Bacteroides and firmicutes, and that extended down through the rectum. So large differences, and I think that points to the fact that maybe we could start to stratify patients in drug trials, probiotic trials, and stuff. That these patients here may not respond as well as these patients, or will respond differently in ways when we're trying to remediate these differences. And these weren't just a few bugs that were causing these differences. There was actually a whole list of particular organisms that were depleted in these dysbiotic subjects. I highlighted here, seem to be, a lot of these were butyrate-producing bacteria, and butyrate is one of the key energy sources to enterocytes, and so these patients with dysbiosis are missing a lot of the bugs that are involved in the secondary metabolisms that actually feed and keep the gut healthy. Others, like Faecalibacterium prosnitii, has been shown in mouse models to be to ameliorate colitis. So if you feed it to a mouse that develops colitis, it won't develop the same kind of phenotypes. And so these are all missing organisms. So you might imagine that Crohn's disease may not be caused initially by this loss of the bacteria, but we're certainly depleting a lot of the bugs that keep the bowel healthy. So maybe propagating or the chronicity of this disease. And so we were kind of interested in that split. So why the different phenotypes or different microbiomes in these different groups? And that kind of led us to what we can do beyond the who's there, and that was obviously hosted environment. So we began to look at hosted genetics and hosted environment through gene expression profiling. At that time, we were introduced to a force of nature named Ellen Lee, who's a MD PhD professor who was at WashU, now was at Stony Brook, who was very involved in genotyping Crohn's patients, and she, and so there's, as I said, about 70 genes that are involved. Two of the more important ones are NOD2, which is involved in innate immunity, pattern recognition, and kind of intracellular pattern recognition receptor, and then it's various genes involved in autophagy. And so we wrote HMP, we were awarded an HMP demonstration project about in 2009 or so to, first of all, genotype the previous cohort, and then also get a much more focused, well-phenotyped cohort together, which was looking at only the ileal microbiota and ileal Crohn's disease, which is a much more steady, kind of easily diagnosed phenotype. We also chose to look at the disease-unaffected region. So before there was just all that destruction and ulceration of the colon, the ileum, would we see differences in the microbiome? And there we've got another large cohort together. And then the third aim was to look at host gene expression in these same tissues where we were looking at the microbiome. And there, now, in just a matter of years, we went from 15,000 sequences to doing full-length Sanger contigs, 500 per sample, and then 454 were just coming online. And so we had all this sequencing done at the Genome Institute at WashU. And we got around 1 million reads from both V1, V3, and a V3, V5 clone libraries. At that point, then, ExploreSeq was completely overwhelmed, and really Chuck Robertson stepped in and really saved the day. That the project wouldn't have been possible without his skill at putting together, under extreme duress, a pipeline that would allow us to take these millions of reads and make sense of them, and also get the kinds of data outputs we needed to move on. And what we found in both of these cohorts were there were significant differences in the microbiome associated with the NOD2 genotype. And so this is from the EPRU cohort. Those individuals who were homozygous for 2 of the risk alleles had significantly different microbiomes compared to those that were homozygous for the non-risk alleles. And then the intermediate heterozygote was kind of an intermediate phenotype there. And then in the WashU cohort, we found that the NOD2 actually had effects on the microbiome independently of disease phenotype. So this is an analysis where we were adjusting for both of these main effects. And so what that means is you may not have, some of these patients actually had not been diagnosed with Crohn's disease, but they had the risk alleles and had some changes in the microbiome. It also means that individuals who had phenotypically Crohn's disease but were lucky enough not to have these risk alleles had different microbiomes. So again, this is quite a stratification in that one kind of remedy may not be efficacious in terms of fixing your disease if you're plus or minus these alleles. And these were really some of the first studies that tied in humans genotypes to differences in microbiome. And so really the summary of this point was that dysbiosis was associated with the NOD2 genotype, and so host does influence the microbiome. This is, these changes we were seeing is not simply the effects of treatment history, diet, etc. At the time, we haven't had power to look at the autophagy genes, but that's something that's continuing. We saw differences in microbiome even in the disease unaffected tissues, so probably means there's large-scale effects of the microbiome that may be driving what was later development of pathology. This dysbiosis was associated with earlier times of first surgery, so we think that's pointing at the fact that the bugs are actually influencing the extent of the disease. I haven't shown you the data, but we also at the time did a whole gene expression profiling of ileal tissues from exact tissues from which we were sampling the microbiome, and found that there were significant differences in pan cell functions. These are specialized cells that produce antimicrobials, and they're also affected by the ATG and NOD2 alleles. And so we found changes in the microbiome correlating with both those and some genes that were involved in xenobiotic metabolism. So there's this dense kind of interplay between host genetics, host gene expression, and the microbiome that is summarized here, where it's probably the loss of the protective microbiota, which is contributing to the loss of mucosal barrier defective function, and you have this vicious cycle now where you've got a chronic disease as opposed to one that resolves once you treat the inflammation. And so we continue to work on IBD. My lab's also shifted focus a looked at a lot of early colonization, how these babies are assemble their microbiome in various circumstances. We're involved in projects looking at mother-to-child transmission through breast milk, and so how feeding practices affect both the microbiome, development of the immune system, susceptibility to HIV. That's done mainly in Uganda. We have a project looking at how micronutrients, which are involved in growth of altering, might affect the microbiome there for better or worse. And then in the early colonization, how that affects susceptibility to infectious diseases. And finally, a lot of maternal child programming. So we know that mothers are one of the great sources of the microbiome initially, but if they have pathologic microbiome, such as associated with obesity or diabetes, are they passing those on to their infants? And so we work with a great group at the university who are looking at, or pros at looking at all the metabolic phenotyping of infants and studying how changes in the microbiome may affect the early development of some of these emerging diseases. And at the same time, we're also continuing to develop analytical tools. That's really been spearheaded by Chuck Robertson and Kirk, and then Brandy Wagner, who's a statistician we work with. We recently released a program called ExpelSet, which is a great way to take your OTU tables and manage them and explore your data sets. And so this is, and then we're also in metatranscriptomics and then developing other statistical methods with some of our other colleagues. And so this is ongoing projects we do, in addition to the more disease-focused studies. And so just to close, I'd like to first of all thank Norm. Norm was very patient with me, let me develop my own slow way, and really guide me. John and Cheryl, thank you for everything in terms of organizing this. Pace Lab, everyone has been wonderful to interact with and learn from. Especially, I think Kirk and Jeff were really selflessly pushing a lot of the high throughput sequencing, barcoding and stuff into the mainstream in the lab. And then the rest of my lab who actually do the work while I sit in my office and fret about grants. And at that point, I'll close and take any questions.
So going back to the NOD2 stuff, most striking thing that jumped out at me was those people carrying the risk alleles. The biggest change that occurs the firmicutes. I mean, there's a huge uptick in that compared to proteobacteria or whatever. And you related that possibly back to the production of antimicrobial peptides, right? And at cells?
I think the opposite. The disease is associated with lesser amounts of those good firmicutes.
Okay, that's backwards.
Yeah.
Well, anyway, is this a gram-positive specific kind of thing? Have you guys looked into what antimicrobial peptides are targeting specifically in that microbiome?
Well, there are risk alleles in the defenses. We know that various of those are produced in those Panna cells. We haven't done those direct studies where I think now we can do anaerobic culture things to understand those. But yeah, those, we know the phenotype, we don't know the mechanism, the chemical mechanism there yet.
Well, the reason I bring that up is there's some interesting work that just came out. A guy named Mike Gilmore at Harvard, they just published a paper in PNAS. They're showing that the commensal organisms of the microbiome, in response to potential invasion orally by pathogenic, multi-drug resistant organisms, start producing pheromones that specifically target the offending organism and kill it. It's a pheromone-based response by the microbiome. So I'm wondering how that plays into that. It's pretty interesting outcome.
Yeah,
we should talk more later.
Maybe just you might come back to that. He's right. The chart on the right, the yellow one, the inverted is yellow. Yeah.
Yeah, this one here. So
Oh, yeah, sorry.
yellow.
I was focused, those bacilli are, I was focused on these Clostridia, the Lactospiraceae, those are all the butyrate producers, so that's, sorry. And so this could be, various other bacilli that are coming up, so, sorry.
Okay.
Yeah, I would focus, my mind is on the butyrate producers, which are these guys down here.
So maybe the more severe disease had more antibiotic therapy?
You know, yeah, I left that off for the study on the right. We had good diet records, we had antibiotic records, we had foul prep records, and none of those were factors here. Now maybe we've got too many variables and we weren't able to really factor those out, but I think the problem is all the Crohn's disease basically had the same suite of antibiotics and steroids and stuff, and so I think there's undoubtedly going to be effects, but we just didn't have that power here, and we didn't have the right set where we had different pre-treatment, and so that was something Norm and I many years ago tried to get a hold of some pediatric samples of just first diagnosis, and that just never got that together.
Does it know what NOD2 does? I mean, it's fascinating to think about think about the host actively molecularly interacting with the microbiome by making a receptor, or making certain metabolites, or bringing some proteins.
So, TLR receptors, which are outside the cells, exposed to the environment, they're involved in responding to damage or molecular pathogenesis. NOD2 is one of those factors. It's an internal signaling factor that as cells are being sampled, brought in, or invading the epithelial cells, it's one of the things that responds to those bugs. It also actually has physical effects as tethered to some of these autophagy genes. So what's the defect here, and it's been kind of controversial too because people publish various mouse models that either truncated NOD2 and have different effects on the microbiome is what we see in humans, and so
Are people focusing on that as a way for therapeutic intervention? Because we understand those processes, that seems like the best way to think about how to manipulate.
I don't know in terms of that. I think there's a lot of emphasis now going on in probiotics and bowel transplants, and so
That's looking at sort of the health of the microbiome, not thinking
Right.
about how
The
the host
host.
is actually
Yeah.
influencing.
I don't know that as well in terms of chemotherapy.
Okay, thank you, Dan.